Hey everyone, my name is Saiteja and my ID is 2014014036. This is a presentation for the course of Elements of Synthetic Biology Life 2.0. Our project is on the topic of CRISPR and HIV. I will be talking about the topic of how CRISPR is used to cure HIV using gene editing methods. Initially for HIV and its infection. HIV, which is known as the human immunodeficiency virus, is a virus that is able to integrate its genome into the host cell genome during infection. HIV primarily infects the CD4 plus T cells and leads to AIDS, acquired immunodeficiency syndrome, if untreated. HIV viral particles store their genetic information as double-stranded RNA and use a reverse transcriptase to convert their genome into a double-stranded DNA. This process also introduces many random errors into the HIV genome, allowing the virus to rapidly mutate. The DNA is inserted into the host cell genome by integrases which use LTR, which are known as the long terminal repeat sequences on the viral genome, to integrate with the host DNA. The result is a reservoir of viral DNA in infected cells, also called a provirus, that can continuously produce new viruses that can infect other immune cells. Currently, the disease can be controlled by rigorous antiretroviral therapy, which are therapies targeting the unique reverse transcription step in the HIV replication process. However, the threat of virus re-emergence is always there. Now coming to the treatment using HIV. Now coming to the treatment of HIV using gene editing. The initial phases of HIV treatment using gene editing and synthetic biology methods included methods like zinc finger nuclease ZFNs, which are artificial restriction enzymes which are used to target specific desired DNA sequences, and that and this enables zinc finger nucleases to target unique sequences within complex genomes. Next comes up transcription activator like effector nucleases. TALEN are these are restriction enzymes that can be engineered to bind to practically any desired DNA sequence. So when combined with a nuclease, DNA can be cut at specific locations. Now, however, these approaches could permit some viral protein production. So researchers were also interested in some precisely excising the HIV-1 genome for infected cells. Researchers believe that using CRISPR-Cas9 could improve efficiency and reduce the probability of off-target effects. Then came up CRISPR. Now using CRISPR-Cas9 against HIV. While using CRISPR-Cas9, the main functions that are expected from CRISPR-Cas9 in the treatment are excising the HIV genome from every infected cell and to prevent the reinfection of those cells. For carrying out this particular function, CRISPR uses the main approach that CRISPR uses in this method involves the alteration or editing of the LTR expression. LTRs are the long terminal repeats which are identical sequences of DNA that repeat hundreds or thousands of times, found at either end of the retrotransposons or proviral DNA found by reverse transcription of retroviral RNA. They are used by viruses to insert their genetic material into the host genomes. Now, coming to the treatment method. The CRISPR-Cas9 system can introduce double-stranded breaks in DNA at a site determined by the guide RNA. Researchers targeted a CRISPR-Cas9 system to target the LTR regions of the HIV provirus at positions T5 and T6. Jurkat cell lines with HIV provirus-like regions that produced GFP instead of the viral proteins were used to determine the effectiveness of the potential Cas9-based therapy. As shown in the diagram, we can see how the Cas9 has been used over the Jurkat cell lines, which have similarities to the HIV provirus lines. Now, the cells were transfected with a plasmid containing the humanized CRISPR-Cas9 and guide RNA genes. Targeting of the LTR by the guide RNA resulted in significantly decreased transcription of the genes under the control of the LTR promoter, as demonstrated by flow cytometry of cells with GFP under LTR promoter control. Furthermore, multiple transfections by the Cas9 plasmid were shown to significantly reduce the transcription of GFP, indicating that the CRISPR-Cas9 system would be effective in preventing the reactivation of latently infected cells. Now, with the sequencing analysis of CRISPR-Cas9 target site, yes, we now have sequencing of the LTR promoter region revealed various mutations at the cut site. These mutation patterns are likely to be the result of the non-homologous end joining pathway as the cells attempt to repair the DNA after double strand breaks were introduced. This indicates the effective cleavage of the proviral genes by the CRISPR-Cas9 pathway. But more importantly, researchers were also able to demonstrate the CRISPR-Cas9 systems and can excise the HIV provirus from the host genome. After 3 rounds of transfection with CRISPR-Cas9 targeting of the LTR, on average the provirus was completely excised from the genome in 31.8% of all cells. A recent paper expanding on this research showed that transfection of macrophage cells with the Cas9 plasmid targeting HIV genes can confer or grant immunity to HIV infection to the transfected cells, and thus Cas9 plasmids could even be used as vaccines against HIV or other viral diseases. Now coming to the hurdles that the this industry faces. Translation of these engineered CRISPR-Cas9 systems to actual gene therapy in HIV patients still has a number of hurdles, namely the efficiency must be quantified and improved off-target effects must be minimized and an appropriate delivery system must be developed. Hugh and his colleagues recently demonstrated that their CRISPR-Cas9 system for treating HIV-infected cells produced no observable off-target effects. They concluded that Cas9 provides a viable approach to curing and preventing HIV infections, as well as other infections, and can provide a personalized approach to specific HIV targeting. Given the small size of the CRISPR-Cas9 system, a lentivirus vector could deliver the CRISPR system. Now, this was the main study of CRISPR-Cas9 treating HIV viruses. Even though this has many kinds of advantages over the fields, there are many improvements which have to take place in order to make a proper human trial. So, recently HIV is coming up with various defense mechanisms where it is trying to overcome many efforts by the CRISPR-Cas9 system. So hoping for the best that we get a cure for this. Thank you.